This is Rizika Batbat discussing the process of mitosis and meiosis. So, what is mitosis? Mitosis is the process in which a eukaryotic cell nucleus splits in two, followed by division of the parent cell into two daughter cells. The word mitosis means threads, and it refers to the thread-like appearance of chromosomes as the cell prepared to divide. And here is the image of stages of mitosis. There are four basic phases of mitosis, which is prophase, metaphase, and anaphase, and telophase. First is the prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus known as chromatin condenses. Second is the metaphase. Metaphase is a stage in the cell cycle where all the genetic material is condensing into chromosomes. During this stage, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell. During this stage in human cells, the chromosomes then become visible under the microscope. Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic materials carried in the nucleus of a parent cell into two identical daughter cells. During anaphase, each pair of chromosomes is separated into two identical independent chromosomes. The next is the telophase, is the fifth and final phase of mitosis. The process that separate the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. Cytokinesis. Cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into two daughter cells. Mitosis in each of the two meiotic division result into two separate nuclei contained within a single cell. So here is the meiosis. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cells by half and produces four gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a cell, the number of chromosomes is restored in the offspring. Prophase 1. Meiosis 1, the first meiotic division, begins with prophase 1. During prophase 1, the complex DNA and protein known as chromatin condense to form chromosomes. The pair of replicated chromosomes are known as sister chromatids, and they remain joined at the central point called the centromere. A large structure called a meiotic spindle also formed from long protein called microtubules and on each side or pole of the cell. Metaphase between prophase 1 and metaphase 1. The pairs of homologous chromosomes form tetrads. Within the tetrads, any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination. Recombination is a process that breaks, recombines, and rejoins sections of DNA to produce new combination of genes. In metaphase 1, the homologous pairs of chromosomes align on either sides of the equatorial plot. Anaphase 1 then in Anaphase 1, the spindle fibers contract and pull the homologous pairs, each with two chromatids, away from each other and towards each pole of the cell. Telophase 1. During telophase 1, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. Each daughter's Each daughter cell is haploid and has only one set of chromosomes. Half the half the total number of chromosomes of the original cell. Prophase 2 meiosis is a mitotic division of each of the haploid cells produced in meiosis 1. During prophase 2, the chromosomes condense and a new set of spindle fiber forms. The chromosomes begin moving toward the equator of the cell. Metaphase 2 and Anaphase 2. During metaphase 2, the centromeres of the paired chromatids align along the equatorial plates in both cells. Then in Anaphase 2, the chromosomes separate at the centromeres. The spindle fibers pull the separated chromosomes toward each pole of the cell. Telophase 2. Finally, during the telophase 2, the chromosomes are enclosed in nuclear membranes. Cytokinesis follows, dividing the cytoplasms of the two cells. At the conclusion of meiosis, there are four haploid daughter cells that go on develop into either sperm or egg cell. And that's it for this discussion. Thank